in the previous video we discussed about we had an introduction about what is DNA and on the four nitrogenous base pairs and how it replicates. Now in this video we're trying to go in a little bit deep of how does actually DNA replicates and what kinds of enzymes are involved in it. So first of all, let's just for the review like the semi-conservative replication, and what it is that the original DNA strand splits up, a new nucleotides come attached into it, and when nucleotides attached to it, so we have two now strands of DNA, half of which have are from the original pair. So this half of the original pair of the DNA is conserved. So that's why we call it the semi-conservative. Now, in this video, let's go a little bit of deep. So before we start, just let's understand some of the terminologies right here and some of the things that you should know in order to begin. First of all is that if you notice something here, as I mentioned in the previous video too, that the strands of DNA are anti-parallel, which means one of them is going in this direction and the other one is going in that direction. And so you might be thinking, okay, how do you know? How are you saying that? And if you notice here it says it's a 5. Oh, let me remove this. It's blocking the. Here it says, look here, yeah, you can see it right now here. It says it's a 5N, and here it says it's a 3N. Here it says it's a 5N, and here it says it's a 3N. So they're opposite. And what do they mean by that? And what they actually mean is that if you count the number of carbons, so you start off here 1, then it's the second one 2, then it's the third one 3. Here is the fourth one 4, and this is the fifth one, the one which is joined with the phosphate group. Now, if you notice this diagram right here, you see that it's calling the fifth end because the fifth carbon right here, is attached to the phosphate group, is on this side, and on this side the third carbon 1 2 3, the third carbon is attached to an carboxylic group. A hydroxylic group, not a carboxylic group. Attached to an hydroxylic group, which is on this side. So these two are opposite. So you think that, and here is the OH group, and so it's going in this direction, and this one is going in the opposite direction, and which really helps in DNA replication. And we will discuss how does it happen. So DNA, when it wants to replicate, let me extend the page. So when the DNA wants to replicate itself, it basically has, so since in bacteria or other organisms which do not have as much big DNAs or as much as big strands of DNA as we do, they usually do it in an easy way. It happens within an hour or so. But when we are talking about a human DNA, it's huge. It's sometimes it's really long. So it would take us a lot of time if we just start from one end and try to go all the way and try to replicate all the strands of the DNA. So what they do is that as DNA wants to replicate, so if I'm drawing the DNA right here, and I'll tell you what these are not these are like some sort of bubbles. So this is one strand of the DNA and this is the other strand of the DNA. So what is happening here is that these strands of DNA have special kind of nucleotides attached into it, where proteins come attached into it and start off replicating from this site, specifically this site. And what happens is that these places where the replication is are called origins of replication. Origins of replication. Origins of replication. And these sort of bubbles, you may view them as these go on the opposite direction, and as they go to this one right here goes in the opposite direction too, and as they do, the bubble extends like this, the bubble extends. So now the bubble is a little bit thinner like this, and it's extending, and you eventually end up with 4 strands of DNA. So it keeps on extending, and eventually you end up with 4 strands of DNA. And so this is actually how the DNA replicates itself. Now, if we look at a closer view, if you look at a closer view, so you might be thinking, okay, why did he mention about this 5 to 3 and 5 and why was he mention about these 3 and 5 carbons, and what's it used for? Well, it's quite useful because when the DNA wants to replicate, when it's first of all, it has to separate the 2 strands from it, and it actually does that with the help of an enzyme called helicase. So helicase is the enzyme which is responsible for breaking the 2 strands, which means that breaking the hydrogen bond between the 2 strands right here. These are the 2 strands. These are the 2 strands, and what's going on? These hydrogen bonds are broken up by a helicase, and as it is breaking them, the new strand is always forms from the 5 prime towards the 3rd prime. And what do you mean by that? I mean that since they are opposite, right? So since this is the 5 and this is the 3rd, and this is the 3rd and this is the 5, so if I were to separate them, so what the opposite of 3 would be 5. So a new strand of DNA would start off from here and go all the way down. It always starts from 5 to 3rd, and its corresponding strand would be 3 to 5. So why does it always start from 5 to 3rd and not 3rd to 5? And hope I'm not confusing you. And why does it do that? Because I'll tell you later. I don't want to confuse you a lot. So just keep in mind that the new DNA, the new strand of DNA always starts from 5 to 3rd. And just for representing a model, I had this picture. Here it is. And oops. So here it is. So this is the one strand of the DNA, and this is the another strand of the DNA, and this is the helicase enzyme which I was talking about. Helicase enzyme, and this model is called the fork model, and this part right here is just the half of this bubble I'm showing you right here. So this half of the bubble right here is representing this molecule. I mean, is representing this structure. So this half of it is representing this structure. Now, as helicase comes and attaches itself to this DNA, it starts breaking away the bonds. Another enzyme. Oh no, sorry. As helicase first enters and starts breaking away the bonds, so you're left with this strand of DNA alone. Now, a new strand of DNA cannot be formed by itself. It must have some starting point in order to start. And how does it do that? Because if you just give a cell, if you just leave a strand of DNA all alone, it won't be able to make a copy of itself, because it must have a starting point. And where does this get that starting point? It gets that starting point from a primer or from a primase, which gives a primer. And what is a primer? A primer is just a small molecule of RNA which it attaches it to the DNA molecule, in order that another enzyme would get, oh, I should start replicating from here. And that's why the replication always starts with 5 to 3 corresponding to this 3 molecule, because in order to give the starting point, or in order to give the starting point or the RNA molecule, this OH group is easily replaced, because since this part right here has an OH group attached to it, it can be easily replaced and the phosphate group, and the RNA molecule can be added into it, or the primase or the primer can be added into here, and the other enzyme which wants to make the copy would get, oh, I should start from here, because here is the primer. So that's why it always starts from 5 to third, and the corresponding to the 5 should be the third end of another strand. So hope that didn't confuse you. So as the helicase is detaching the strands, making them single stranded, some proteins come attached. They call the binding proteins, which come and attach themselves to the strand right here, in order that they don't wound again, and they just keep them unwound till the replication process occurs. So a primase comes here and here it is the primase right here, the red one. This is the RNA molecule, which comes and attaches itself to the DNA molecule at the third prime carbon, which means it has a carboxylic group which could be easily replaced by the RNA molecule. And as it occurs, and another enzyme which is one of the most key important enzymes in the replication process, is the DNA polymerase. At first, when it sees there's an RNA right here, the DNA polymerase comes attaching itself, and as it's going, you can view it as it's sliding down all the way down here. It replaced this part with the RNA with the DNA, and as it's going down and down and down, it's adding nucleotides to it. Now, but you might be thinking, okay, this part of the strand, which is called the leading strand, and this one is called the lagging strand. So you might be thinking, okay, the leading strand can do that. Alright, it's no big deal. This is the third part. It has a carboxyl group which can be from which an RNA molecule or the primer can be easily attached to it, and then the DNA polymerase can come along and go all the way through, adding nucleotides to it and making another copy. But what about this one? Because if you notice, this one is 5. It is the fifth prime, which means it has a phosphate group attached to it. It cannot easily make a copy of itself, because it should go from where to where? Well, I just mentioned it should go from 5 to 3, but here is 5, so the opposite one should be 3, but which is not possible. Well, you're right. This is hard for the DNA to do that. So what it does is a really amazing process. It's that another primer, a starting point comes along. Here it is, the RNA primer. It comes along, attaches itself here, and as it attaches itself, the DNA polymerase it starts from this primer and starts from this primer and goes all the way, making a strand, and this leaves. And as the DNA so now you have a DNA, but it's working in the opposite direction, which means it's going from 3, which means it's not going in the opposite direction actually, but it's doing the same thing but in the reverse process, which means that this is 3, but it gives the primer over here, so that the DNA polymerase starts from 5 to 3. Then the DNA polymerase leaves. Then after a few steps, the primer again comes and gives another primer. So this one becomes 5 and this one is 3. So it again goes this way, but it's going from 5 to 3, but it's going in the opposite direction, which means the DNA polymerase is doing the thing which it has to do. It's making the duplicate molecule by 5 to 3, but it's simple. And if hope I'm not confusing you, but the thing is that if I were to draw this part right here, since it's 5, I can't go this way. So what happens is a primer or an indication comes. So okay, start from here. I said okay, if it's 5, so this one should be 3, right? And if this one is 3, this one should be 5. So the DNA polymerase starts from here and goes all the way down, makes a DNA strand, a segment of a DNA strand, and then just leaves the DNA molecule. Then after the DNA has been unbounded more, another primer comes along. And this time this one is 5 and this one is 3. So it's again going from 5 to 3. And then as it's going down, it's leaving the nucleus again. And so this process keeps going on. So you can imagine that the direction of the process is here, but the 5 to 3 is going in the opposite direction. The process is going along with the DNA strand, which the leading strand is going through. So these small fragments of DNA which are being made here, so you can see from this red one to this another red one, this is a small fragment of DNA. These are called the Okazaki fragments, named after the scientist Okazaki himself. Okazaki fragments. And these are just basically small segments of replicated DNA. And what happens is that in order to attach them, these RNA, as DNA polymerase comes, it removes these RNA and makes the whole strand DNA, and it leaves the nucleus. But there are still, so these fragments are joined to each other. So this fragment and this fragment are joined to each other. So these two fragments, like these are, which are separated by these primers, are joined to each other by an enzyme called the ligase. So ligase enzymes joins the Okazaki fragments to each other in order that they join together and form a full strand. And this strand is called the lagging strand. So this is the mechanism how which DNA replicates itself. And just for a quick review, helicase comes along, breaks the hydrogen bonds between them, and these proteins, SSB proteins, join these strands of DNA in order that they don't bind together again. And again, a primer comes along. It gives a starting signal. Why? Because the one strand of DNA cannot duplicate itself all alone. It must have a starting point. It gives the starting point. The DNA polymerase comes along. It makes the whole strand of DNA from 5 to 3. Why 5 to 3? Because opposite to 5 there is a 3' carbon molecule which is attached to a hydroxyl group to which the primer can be easily attached. It means the starting point can be easily given. So that's why it starts from 5 to 3, and it always goes from 5 to 3. But the other strand, the lagging strand, has a little bit of difficulty because the main strand molecule starts off with 5. So it should do the opposite, but it doesn't do that. So what happens is that another primer comes along and gives it the signal here, like this is the 5 carbon molecule. This is the part of the molecule. You should start from here. It said, okay, so this is my 5' carbon, and it makes the strand, a segment of the strand, this direction right here, and it leaves the nucleus. So there's a primer right here, and after a little while, there is another primer, another starting from. Okay, start from here. He says, okay, the DNA polymerase comes along, goes from 5 to the 3rd carbon, which means this one is right here is attached to the hydroxyl group, and this one is attached to the phosphate group, and it makes another small fragment of DNA and leaves the DNA. Now these small fragments are called Okazaki fragments, and in order to join them, ligase comes along and removes these and join the whole strand together. And this is how the molecule DNA replicates itself. And hope you found that useful, and see you in the next video.